All right, so first off, I'd like to thank Davo and the ISHR leadership for offering this great webinar series. It's really been terrific to get to sit down over lunch and watch a great webinar, good science on my TV, as at least one of the better aspects of this new normal. So for a long time, I've been fascinated by the very amazing different ways that biological systems can remodel themselves. In the case of the heart has a variety of ways to remodel its size and shape in response to different stresses. So for example, as you see like a normal looking heart here, in the case of chronic exercise or pregnancy, you can get hypertrophy, an increase in size of these cardiomyocytes. And with these more physiologic type of stimuli, this is reversible. However, in more pathological forms of hypertrophy, as we see in hypertension and stenosis, we see a more pathological form that's accompanied by fibrosis, is more irreversible. And for reasons we still don't quite understand, this can rapidly switch from a more concentric remodeling to a dilation of the heart and sends you downward in one of the spirals of heart failure. In contrast, other types of stimuli, such as myocardial infarction or gene mutations associated with dilated cardiomyopathy, can cause eccentric remodeling and dilation. So I've been fascinated by the various ways in which this heart can remodel, especially in terms of the molecular mechanisms that may be controlling this. And so with this in mind, my lab in systems biology is interested in identifying the molecular networks that determine the control points that decide how the heart responds to stress, and with the goal of using this understanding to work towards therapeutics that might control or pull the strings on these molecular networks to prevent or reverse heart failure. We've been particularly interested in biochemical or mechanical stresses that cause decision-making in the heart, where our early work focused on contractility and arrhythmia, and increasingly we've been working on hypertrophy, fibrosis, and proliferation. So today's talk will be focusing on our work on fibrosis. As David mentioned, I do come from an engineering background, and so we try to use like an engineering spin or engineering guided approach to both our modeling and our experiments. We develop computational models that piece together different aspects of what is known about these complex systems, make predictions that we then validate with the quantitative experiments. And in cases where we have a rather incomplete understanding, we iterate this back where we infer the biological mechanisms from these quantitative experiments and use those to develop computational models that represent hypotheses of how that system might be working. So then focusing more towards the fibrosis aspect, wound healing is a really complex process in myocardial infarction and in other cases that represent a really delicate balance of extracellular matrix synthesis and degradation. Ideally, we have a progressive and controlled transition from an inflammatory phase to a reparative phase to maturation and a stable patent scar. However, there are a lot of things that can go wrong, such as increased inflammation that can lead to rupture, dilation, systolic dysfunction, or if you have too much matrix deposition, you can get exacerbated fibrosis and diastolic dysfunction. Both of these can kill you right off the bat with arrhythmia. So there are many things that could go wrong in this situation. And because of this, there's really a challenge in developing therapies that would need to have very fine-tuned control, especially given the substantial patient-to-patient variability over how these processes proceed. Another challenge in therapeutic targeting is that there is a tremendous complexity to both the cell types, such as myocytes, fibroblasts, neutrophils, monocytes, as intercellular signals and cytokines and different growth factors. And also, when you look inside the cell, there are many different signaling pathways, transcription factors, receptors that are controlling these events. And so this complexity really hinders our ability to both pull the field together and understand how this works as a system, and also to quantitatively prioritize one target over another. And so with these challenges in mind, our group has been motivated to develop computational models that might help integrate some of these mechanisms to systematically identify drivers of cardiac remodeling, in this case, myofibroblast activation and matrix remodeling. So our first step in this direction was led by Angela Ziegler, a very talented MS2P or MD PhD student in my group. And she started with a very systematic literature survey to reconstruct the known signaling network that regulates cardiac fibroblasts. And so what she did is very detailed curation of the literature, making sure that we had at least 2 citations per reaction with an emphasis on cardiac fibroblasts. And so she reconstructed this network composed of about 130 reactions from 177 papers. And so this includes different pathways or modules such as cytokines, TGF-beta signaling, G protein signaling, and stretch signaling. And then these receptors at the top converge down to, in part, transcription factors that regulate gene expression. We have very different various outputs of this model, such as the different collagens, fibronectin, periostin, fibroblast proliferation, and also extracellular signaling to some extent in terms of regulation of MMP activity. And these MMPs can degrade various peptides and activate some of them. So far what you're seeing is really like a detailed literature survey that is similar to what you might see in a review article. And so what we next want to do is to see what can we do to actually make mechanistic predictions from a network like this. And so in order to do that, we need a modeling framework. And so I'd like to now introduce you to a method that we developed about 10 years ago called logic-based differential equations. And the basic idea is a really simple one in that, say if A activates C, this A could be a protein, a gene, a small molecule, you name it. We're going to make a series of very simple, basic assumptions that are honestly a bit phenomenological, but hold with me for a second. We assume that A activates C with a saturating or sigmoidal type relationship. That's actually quite common in biology. And then further, we assume that the timing by which A activates C happens according to this time constant tau. Second, we're going to make some assumptions about, rather than tracking the absolute concentrations of A, like 10,000 copies per cell, we're going to look at it in terms of fractional activation, where 0 would be not active and 1 would be fully active. We can have a activation value anywhere on this axis, so this continuous, not Boolean. Further, when we have crosstalk between pathways, we assume that that can be either an AND type logic, where in this case X and Y affect Z multiplicatively, or an OR gate type logic, where either X or Y would be able to activate Z. And then finally, we do have some parameter values in this model formalism, but we're going to do something really different than what we had seen before, is that because those parameter values are rarely known on these large networks, we're going to make a significant assumption and just set them to default values that are consistent across the network, where, for example, in the timing of these different reactions, I'm going to say signaling happens on the order of about 10 seconds, transcription happens about on the order of 1 hour, and translation happens about on the order of 10 hours. So with a set of really basic assumptions and a very simplified modeling approach, the next question would be like, well, is this good for anything? And so to evaluate that, we went back to this detailed biochemical model we had developed that David had mentioned of beta-adrenergic signaling. You have the beta-adrenergic receptor here going down to pKa and its regulation of different ion channels. And we performed a sensitivity analysis that basically represents what would happen if you could knock down every member of this network and measure the response in every member of the network. So that'd be a huge experiment, but would give you a really comprehensive idea of how things are functionally related in the network. In this case, this model had been developed from about 90 parameters from the literature, so representing multiple decades of data being integrated and required to run these simulations. In contrast, we could develop a model using the new logic-based formalism of the same exact network topology, just without any of the parameters. And what we found, surprisingly, was that the functional predictions that this model could make were actually quite similar, despite not using any parameters from the literature. And so this gave us confidence to then use this new approach to model systems that could not be modeled by traditional biochemical approaches. And so over the last few years, we've taken this approach to model a variety of different networks in the heart, such as the cardiomyocyte hypertrophy signaling network, a mechano-signaling network. This is the logic-based version of the beta-adrenergic pathway, and most recently, a macrophage activation network. All of the work that I've been telling you about, or will be telling you about, uses this software that we've developed that's open source called NetFlux, that allows you to automatically generate the equations behind the networks. And so rather allows you to get very systematic model development process. So now let's turn back to the fibroblast and take a look at what happens when we use this approach to run simulations on the network. You can run virtual simulations in these models now with the logic-based differential equations to predict the dynamics by which everything within this network would change in response to a particular stimulus, such as adding TGF-beta right here. And so here we're seeing, after we add TGF-beta, we're seeing increase in collagen expression, alpha-SMA activation, and autocrine production of IL-6 in endothelin, for example. So this shows that we can build something really big and run some simulations on it. It doesn't yet give us any confidence that this model is worth anything. So of course, in that case, we need to perform a lot of validation comparing the predictions of these models against independent sets of experimental literature. And so we did an awful lot of that. Let me just start with one example, and then I'll show you how that generalizes. So for example, in this case, if we add TGF-beta to the network, we get a response in collagen, alpha-SMA, MMP activities. And then we follow that by adding forskolin, which activates adenyl cyclase and produces cyclic AMP. And the model predicts that if you chase TGF-beta with forskolin, you suppress the effect on collagen. And similarly, data from Paul Inzel's group shows that the same situation occurs in that TGF-beta, of course, increases collagen production that can be subsequently suppressed by forskolin. And so this shows the type of qualitative agreement that would give some level of confidence in these model predictions, because this type of data was not used in building this network. We've done that in a lot of different cases. These little charts right here are summarizing data from about 34 different papers where we found an overall validation rate of about 80% against independent data from rat cardiac fibroblasts. So now using this model, we can make predictions about things that we don't have data for already. But one thing to point out first is that that model was based on these default parameter values. And so of course we want to know how robust the predictions are to those assumptions. And so we can randomly vary the parameter values, and we find that the validation accuracy is actually rather robust despite uncertainty in those parameters. And so that would make sure that the model is not overly fragile. So now we can run really big simulations and kind of find new relationships within the network model. And so this matrix right here basically represents what would happen if you could knock down each of the 98 different proteins or nodes within this network, and then systematically measure the response in everything else in the network, which would be a huge experiment. And if you think about this in terms of biological terms, this column, for example, would represent what would be the expected response if you knock down the TGF-beta receptor and did like a proteomic experiment to measure the response in everything else in the network. Or this row might represent what if you had an alpha SMA gene expression reporter and you used a knockdown library where you knocked down every gene in this network and looked at its effect on alpha SMA expression. So as you can see, those would be two very big experiments that would contain only a portion of the information encoded here. So you can use this information in a very targeted sort of cherry-picked way, but we're interested in particularly using more data-driven approaches to extract information from these networks in a less biased way. And so what we did is we took that network in the context of a high stretch situation, because we know that stretch is really important for activation, but it's not fully clear why. And then we just clustered the sensitivity matrix. And from that, we identified a number of network modules or sets of these nodes that work together. So we classify these nodes by that, and we then from that could build a simpler representation of the functional relationships within this network. And one of the surprises here to us is that in a high stretch context, of course, the mechanical module was leading to fibrosis, but also we are seeing induction of this autocrine module and TGF-beta signaling in that context that we were not expecting. And so with the model, you can do a lot of follow-up simulations to understand how that's working. And after a bit of analysis, we found was a real surprise to us is that stretch was inducing alpha-SMA expression. That's one of the main markers of myofibroblast activation, not through any single linear pathway, but by activating the transcription factor AP1, which was producing latent TGF-beta, which was getting activated. TGF-beta was then acting in an autocrine loop down through canonical signaling through SMAD3, and then inducing alpha-SMA expression. And so in bar graph form, you can see that the model is predicting that stretching the cells would induce alpha-SMA, and then we simulated a knockdown or an inhibition of the receptor, which we found to suppress the alpha-SMA, showing that this is a very important pathway that we hadn't expected before. And so we want to know, of course, is that well substantiated? And so we then did experiments to validate those predictions. And so this is in a 3D culture of rat cardiac fibroblasts that were either in mechanically restrained gels, so that the gel is kind of harder for these fibroblasts to pull against, or free-floating gels, which are easier for these cells to contract. And so what we see here on the right is that the free-floating gels with the fibroblasts, you don't see so much activation. If there's a mechanical restraint, you get induction of alpha-SMA, and you see these fibroblasts getting kind of spreading out. Whereas if you have a TGF-beta receptor inhibitor, then that suppresses that activation. And we see similar patterns of response in terms of the size of these gels, showing how much contraction happens. And so this provides an experimental validation of this model predicted TGF-beta autocrine loop in mechanosignaling. So then another question would be, is this relevant to an in vivo situation at all? And so while we were not involved in this study, we were really happy to see come out then is this paper from Jeff Mulkenton's lab, where this was in kind of some of the early examples of being able to do fibroblast-specific knockdown in vivo. And they found that knocking down the TGF-beta receptor suppressed interstitial fibrosis in response to aortic constriction, much quite analogous to our predictions of how inhibition of the TGF-beta receptor blocks stretch-induced fibrosis. So what I've shown you so far is looking at an average cell, but we know that there's a lot of single cell heterogeneity. And so we've been interested in scaling up these single cell models to look at what happens in fibroblast populations. And so we can do that by combining these sibling networks with agent-based models, where these triangles represent individual fibroblasts migrating in a, honestly, kind of an artificial two-dimensional environment, where over towards the right, we have more inflammatory cues, and towards the top, we have more fibrotic cues. And so as you saw in that simulation, hopefully, the model would predict that we see the most collagen deposition up in this quadrant, where we have high TGF-beta, but less inflammatory cues. Because it's a multi-scale model, our model can predict not just what's happening phenomenologically in terms of collagen, but also what's happening in terms of intracellular signaling that's happening inside those cells. And so within this spatial environment that we're simulating, for example, we predict, of course, higher IL-1 beta receptor activity, where you have more of those cytokines, but we see quite interesting patterns of response in terms of alpha-SMA up in this area where we have high TGF-beta but lower interleukin-1, and quite distinct patterns of NF-kappa B activity, which is happening in all of these regions except that down here. So it shows that this can be activated by either the fibrotic or inflammatory inputs, and the response is quite switch-like. So we get some interesting patterns of response. And then we can also follow the trajectories of individual cells within the simulation to get a glimpse of the single cell heterogeneity. And so on the top left here, this is where like one fibroblast is starting out in a high TGF-beta environment, and it's going to walk and randomly migrate over into this area of higher inflammatory cytokine environment. And so you can watch its single cell trajectories, for example, here. It's starting out with higher TGF-beta and gradually walks into an area of higher IL-6. And that's accompanied by a high expression of collagen early and MMP activity a bit later. In contrast, this cell over here, it's starting off in a high cytokine area and randomly walks into over in this quadrant. And so it has a distinct pattern of response here. We have not yet experimentally validated the single cell dynamics, but we're really excited about those types of experiments coming out now that would be able to validate these types of predictions. What we could predict is the more type of like bulk responses and crosstalk between the fibrotic and inflammatory responses that this model was predicting. And so as an example of that, I showed you how the model predicts that, of course, TGF-beta will induce collagen expression. And we predicted that combining that with IL-1 beta would suppress the collagen expression somewhat. And we'd also see that at the level of alpha-SMA expression. And in this case, the model did not predict F-actin to be suppressed by IL-1 beta. And when we do similar experiments in human cardiac fibroblasts, we see overall similar patterns of induction, where looking at alpha-SMA, for example, it's induced by TGF-beta 1. And if you look over here, we see that bringing on IL-1 beta suppresses the alpha-SMA activation of these fibroblasts. And there's similar patterns of response to the levels of procollagen and filamentous actin. So what I've shown you so far are all examples of predictions that we're making in vitro. And so, of course, we want to know how that might relate to what's happening in vivo in the heart. And so to take that step, we've taken experimental data from the literature of time courses for a variety of different cytokines from the post-MI literature, and use those as inputs into the fibroblast signaling network model for these 9 different cytokines. So these dots are experimental data from the literature, and then we fit these exponential curves and use those as simultaneous drivers to the fibroblast network model. And so this is the sort of network-wide response that we predict from that. We say very early on we saw the cytokine activity. About a week out, you see very strong TGF-beta signaling, and then a lot of the TGF-beta signaling is kind of gradually coming back down a bit at these late times of post-MI. Again, when you see these predictions, we want to know how good are they? To what extent does this give us confidence in these predictions? And so when we look at independent experimental data for collagen transcripts, like collagen 1 and 3, we see that, like in the experimental data, the model predicts a transient induction of collagen transcripts, and then subsequently a slower, more monotonic rise in collagen area fraction. That's the level of interstitial fibrosis within the heart. And so this shows us that, at least to some extent, the model is able to predict that the differential dynamics of what's happening in terms of matrix expression at different periods after myocardial infarction. So now with this model, we can start to predict kind of more newer relationships over bigger screens in the network model. And so in this case, what we did is analogous to what we had done in vitro simulations. We're gonna, in the situation of predictions of myocardial infarction, we're gonna make predictions of what would happen if you overexpressed a given gene within this network, and repeat that for all of the genes in the network. And we see a variety of really interesting patterns. So a lot of the genes down here are predicting that there's increased fibrosis if you overexpress those in the context of myocardial infarction. These genes over here are predicting that their overexpression would suppress fibrosis. And we also have some really interesting ones in the middle here related to NF-kappa B signaling and IL-6 signaling that are predicted to have biphasic effects on collagen production. And so when we compare some of these predictions against the literature, we find that in cases like SMAD7, natriuretic peptide, NOX4, and CJUN, that these models are consistent with independent experimental studies. But there are cases in which there is not agreement. And one interesting case is the study from Jeff Mulkenton and Jen Davis showing that MKK6, an activator of P38, induces fibrosis. And so when we see these areas of discrepancy, what that means is that there are likely pieces of missing information where we don't fully yet understand how P38 regulates fibrosis, and that provides an opportunity for follow-on studies to delve into those mechanisms. One particularly interesting prediction in here was the role of IL-1 signaling, where the model predicted different effects depending on how strongly you overexpress IL-1. So for example, so this is just showing different levels of overexpression. And then on the bottom here, we're showing what happens at the level of collagen transcripts. And so for 5-fold overexpression, we predict an augmentation of the amount of fibrosis during this 1-week period. But with a higher level of overexpression, the model predicts that that's sufficient to induce fibrosis even without the myocardial infarction, and further that it might suppress the TGF-beta induced fibrosis that would be happening here, but overall lead to long-term fibrosis. And so the model predicts that this some multi-phasic relationship is due to the fact that there are multiple pathways by which IL-1 leads to fibrosis or modulates fibrosis. On the one hand, we have IL-1 through MAP kinase signals driving collagen production, but on the other hand, there are negative regulatory pathways, including BAMBI, NF-κB, and IL-6, that are suppressing fibrotic responses. So we're then curious about whether these models might be useful in predicting how drugs work. And so what we did is we created a new framework for integrating data from drug-target interactions, such as from drug bank, into these network models. And while, as you may have seen in pharmacology or in medical school, there's been a very long literature on how to represent drug interactions with biochemical kinetics. This hasn't really been done in these logic-based frameworks before. And so what we did is we had to do a new formulation that captures the same types of behaviors, like left-right shifts with competitive inhibitors or activators, or up-down sort of shifts, or changes in magnitude with non-competitive activation. And so using the information in these databases and literature, we were able to map 33 FDA-approved drugs onto this network model, and then look at what would happen when we simulate these various different drugs. One thing that's interesting here is that there are multiple drugs here that have the same target but different binding modes, and so that could give interesting predictions. And another interesting thing is that some of these drugs have multiple targets, and so it provides a chance to use the model for polypharmacology. And so we simulated the response of these various different drugs in the context of myocardial infarction. And what you can see here on this left heat map is at different times post-myocardial infarction, the model predicts different responses for these drugs. So for example, with Entresto and sacubitril, it's predicting really suppressing the collagen that's produced over that period. Whereas, for example, arsenic trioxide, which is used sometimes as a cancer therapeutic, is predicted to be pro-fibrotic at all these times. And they're somewhat similar, but actually, somewhat distinct patterns of these drugs and how they affect different aspects of fibroblast function. For example, this is showing the proliferative response. And we've now been looking at to what extent these predictions are consistent with the literature of drug development. And a variety of these are consistent. For example, the model predicts a suppressed collagen synthesis with cyclic GMP, which is produced in response to nitrates. These predictions for arsenic are consistent with this study from Chu et al. These predictions for Entresto, which is a combination therapeutic of sacubitril and valsartan, the model is predicting anti-fibrotic effects consistent with this study by Loiter. But now remember that there's a lot of mechanistic underpinning to these models. And so it's not just strictly the predictions themselves. There's a mechanistic prediction. And so we can now dig in to look at the network mechanisms by which these drugs may be modulating fibrosis. So in the case of arsenic, it was quite interesting in that the model was predicting that, so the arsenic is activating AP1 signaling, and the model predicted that it's doing kind of two things that work together. First, that it's inducing this TGF-beta autocrine signaling by this pathway, and it's also inducing autocrine endothelin production by this pathway. And that it turns out that the model is predicting that these work together to enhance the overall collagen accumulation by a combination of SMAD3 and SRF signaling. And so over here on the top right, the model is predicting the fibrotic responses to arsenic, and then predicting what might happen if we could knock down endothelin or TGF-beta, and that would suppress these autocrine loops. Further, these predictions are at least consistent with separate measurements by that group showing induced TGF-beta expression and the ability in vitro to block collagen expression due to arsenic with a TGF-beta receptor inhibitor. Looking at the case of Entresto was another interesting case study. As I showed you on the last slide, the model predicts that Entresto would suppress collagen after MI. That is consistent with independent studies from the literature. And the model predicts that there are different reasons for this efficacy at different time points post-MI. And so at 1 week post-MI, the model is predicting that the main effects are by suppressing SMAD signaling through cyclic GMP, whereas at later time points, not shown on this graph, it's acting more through the effects on the angiotensin receptors. So we're also interested in doing our own experiments to validate the predictions for these network models, of course. And so these are a few very recent examples that we made looking at some other drugs that have been of interest. So the model, in this case, we're looking at the effect on F-actin. And so looking at the model's prediction that the ROCK inhibitor Vasutil would substantially suppress filamentous actin, and that we predicted a drop in filamentous actin with valsartan as well. And so we did these experiments in rat cardiac fibroblasts, and you can see representative images here at the bottom showing that, like the model predicts, Vasutil does bring down the filamentous actin quite a lot, but there are some things that are not the same as what's happening in this experiment. For example, that BNP was more efficacious than we expected, and valsartan, it wasn't statistically significant, but there may be kind of a partial effect here. And so this indicates that there's some reason to be interested in Vasutil, but this also points out areas such as BNP signaling that are not fully understood or incorporated into the network model. And then for those same experiments, looking at alpha-SMA expression, which is one of the main markers for myofibroblast activity, we see that similarly, Vasutil is suppressing alpha-SMA expression that's seen statistically in these experiments and in the model. But kind of there's a flip from what we saw from looking at filamentous actin. The model is predicting that BNP is more efficacious at regulating alpha-SMA, whereas experimentally we're seeing less of an effect there. So these provide some insights into areas where we'll want to look into how BNP and ROCK signaling may be kind of similar to what we've got in the model right now, but there are some additional revisions that will likely be needed. So to summarize what we've been talking about today, we've been developing these network models of how the fibroblast works, making new predictions, and in one case about how stretch-mediated activation works through an autocrine TGF-beta loop, validating and building computational frameworks to simulate in vivo signaling in these fibroblasts and how drugs may work in diseases such as myocardial infarction. And so using that, we've started virtual screens for FDA-approved drugs, and then used the model to look at the underlying network mechanisms that may be mediating these effects. For example, for arsenic trioxide, Entresto, and doing our own validations for other drugs such as Vasutal and BNP. So with that, I'd like to thank the fantastic group that I've had the pleasure to work with in these studies. This is from our lab meeting this morning. A lot of these studies have been led by Angela Ziegler, working together with an undergraduate student, Ani, and the agent-based modeling had been led by Michaela Ricard, and others that contributed that I mentioned into these studies are Anders and Steve. So with that, I'd be happy to take any questions. Oh, also collaborators is Jeff Holmes and Shane Pierce-Cutler.
Okay, thanks very much, Jeff, for giving such a lucid and engaging presentation. I can see you've got lots of questions already, so can you see them?
Opening.
So perhaps if you just read out the question, then answer it.
So the first is from an attendee. Could you please share the slides? Yeah, it'll be on the YouTube recording in a day or two, whenever that's ready. Would conditional, this is from Junhoi's son, would conditional ablation of some fibrosis-related genes, such as TGF-beta or SMAD, be applied to prevent or treat stress-induced fibrosis hypertrophy? That's an interesting question. So I think there's been a major advance in the basic science about the ability from Tallquist and Mulkenton to genetically target fibroblasts specifically. However, I'm not quite sure to what extent that is most valuable as a basic science tool versus a therapeutic approach. I think there definitely are some situations in which these drugs, some of the drugs that we're talking about, would be really bad in terms of their systemic effects, and it'd be quite beneficial to specifically target the fibroblasts. From Angelo Calderon, can you please provide what is your definition of a myofibroblast? Furthermore, alpha-SMA is not a consistent marker of a myofibroblast in vivo. In vitro, alpha-SMA is activated when fibroblasts are plated on plastic. Yeah, so the use of alpha-SMA as a marker for fibroblast activation is certainly imperfect. It's still the one that's most widely used. And so every time I see people criticize it, I don't also see them not use it. It's just, I think, helpful to complement it with other measures and to hopefully describe what it is that you're actually measuring. And so
yeah,
in those...
Just a question, if you don't mind.
Yeah.
What other measures would you recommend, actually, for activation?
So one of the most sensitive that we've seen is the spread, so like the cross-sectional area of these fibroblasts. And also what you quantify about alpha-SMA is quite important. So with the measures that I talked about and was most typical, we're just looking at, is there expression? But another measure is, to what extent is the alpha-SMA incorporated into stress fibers and co-localized with filamentous actin? Okay. And so, you know, we're a bit, well, a few years in, but newer to experiments on this. And so we started with similar measures that are similar to what others have been doing. But as David mentioned, we do a lot of like high-throughput microscopy, and so we've been developing automated measures to look at myofibers or stress fibers within these fibroblasts, things like that. So that's a good point. Oh, and then also this thing about them being activated when plated on plastic. Yes, that's an important point. So that's why in the first study that I talked about, we put them in a 3D collagen gel. And when they're free floating, then they're not activated. But when we mechanically restrain that gel, they activate. In contrast, the last experiments that I showed are on plastic, and so they're mechanically activated, but we actually use that as a benefit in that they're already activated. And so then we're using drugs to test to what extent can we reverse that. But that may actually explain why there's only partial effects with those drugs.
I guess a broader question is the activation through that, Young's modulus of the plastic that they're cultured on the same as the activation through TGF-beta, or other stressor? And actually, do we know that?
I mean, there are a lot of ways by which mechanical stimuli can modulate these cells. We have found, the experiment that I showed is that blocking the TGF-beta receptor suppresses the activation. And in our hands, blocking the TGF-beta receptor is by far the most reproducible, strongest thing that you can do to suppress activation of a fibroblast.
Okay, thank you.
Yeah. Okay, anonymous attendee, will this kind of signaling model vary if you're looking at multi-cell system in vitro like an organoid model? Would multi-cell system prevent you from looking at TGF-beta activation by stretch or TGF-beta release from other cell types rather than fibroblasts? Yes, that's a really good question. So, so far we have not combined the different network models we've developed to look at multicellular simulations. In our simulations of myocardial infarction, instead, what we did is represent the myocytes and macrophages and neutrophils implicitly by using experimentally based trajectories for the cytokines that they produce, and also using macrophage produced MMP activity for the collagen protein. Yeah, we're definitely interested in looking at intercellular signaling in detail, but that requires some very carefully done co-culture experiments that we haven't done yet. Sorry, that just switched to the bottom of my chat list. Let me scroll back up.
So we've seen how to go next.
Yeah. But just specifically, yes, other cells are making the TGF-beta as well. In the case of infarction, I think the literature would largely say that macrophages produce most of the TGF-beta, but I'm really not so sure about that. When we look at like RNA-seq levels from isolated macrophages or TGF-beta, that they're quite comparable. And so I think there's a lot of evidence to suggest that fibroblasts do make considerable amounts of TGF-beta. Okay, from Sina Hadapur, earlier you showed that the model does not accurately predict 16 of the 82 pathways that were tested. Have you taken a closer look at these to understand why they weren't accurate? Thank you for asking the question that way. Yeah, so basically the way I look at this, and I think this is maybe clouded by me coming from an engineering background, is as long as these models make pretty good predictions on a first pass, then that's a framework with which we can understand the failures. And so yeah, that's absolutely the type of thing that we're doing now, is going back and looking at what systematic revisions can be done. Now that's a really challenging problem to think about all the configurations that might be different. And so to do that, we've, like Ali Khalili Maybadi, a postdoc in my lab, has been looking at a lot of developing numerical methods to very comprehensively look at all the iterations of these models in parameters and structures and see, like, look at alternate hypotheses. But yeah,
can I ask a question, Jeff?
Yeah.
So I'm amazed that it works as well as it does, because I'd have thought that, I mean, the exact gain of any step and the time constants would critically determine how stable any of the signaling is.
Absolutely. We were shocked when, I mean, this was 10 years ago now, but yeah, we were shocked that it worked at all. Yeah. And so now that we have these models, then we can understand why it works very, very comprehensively. And so this is more of a philosophical thing, but now in the context of having previously developed these really detailed biochemical models, this question of, well, why does this work? And at an abstract level, I think it's that an awful lot of the concentrations and rate constants are right in the middle of the dynamic range of these responses. And so that's kind of like the fundamental assumption that we're making with this approach, is that we just assume that it's in the dynamic range in a sensitive regime, rather than having to experimentally measure those parameters and then see if it's in a reasonable dynamic range. From Lou Roderick. Hi, Lou. Many signaling pathways are regulated by feedback loops. How do you factor these into your models? And have you tried mapping pseudotime expression profiles from single-cell RNA-seq onto your data? Yeah, so it's a nice questions. Our original hypertrophy models actually didn't have any feedback loops. Not because we didn't think that they're there, they're just totally underreported in the literature. And so it was kind of a question of, well, based on what people are reporting, what can we predict? And it did pretty well. And then so we go back and revise and try to find new loops. In the case of the fibroblast literature, there was more information about autocrine signaling. And so that's what motivated putting in TGF-beta and IL-6 and endothelin autocrine loops, such as that. So they are in there, and there are other internal feedback loops as well. In terms of looking at pseudotime expression profiles, yeah, that's part of what we're doing in the case of myocardial infarction. We have not done that at all for single-cell RNA-seq data, although that'd be, my understanding is that single-cell RNA-seq is particularly good at defining cell types and not quite as good at quantitatively profiling dynamics of an individual cell type, but that'd be a really interesting area to explore. Anonymous attendee, can you use newly identified genes and proteins by novel sequencing to predict how they can modulate fibrosis using these models? Yeah, so we're really excited about that opportunity, like as new things are discovered, to integrate that into these frameworks to make new predictions of how they mechanistically work. And so one example, we haven't done that or published on that in fibrosis, but in hypertrophy, we collaborated with Alan Karma and Jim Weiss, who had done the HMDP, the Hybrid Mouse Diversity Panel. And so they had done gene expression profiling for about 100 mouse strains, identified some genes that were correlated with hypertrophy. And then we did an analysis looking at mechanistic linkages between some of their correlated genes to the network and found some really interesting ones and validated predictions of HES1 being a novel regulator of hypertrophy. That was published in 2018. So that was, to me, an exciting example where we can really use that to motivate why you would prioritize study of a new gene and mechanistically link that to known players. Because it's unlikely that all of these new genes we're hearing about are completely unrelated to everything else we know. Yep, you're muted.
I'd be very curious, Jeff, just to see with the model, a method that we presented in one of the webinars, Matt Ackers-Johnson and myself, of the neonatal hypertrophy model. I'd be very curious to see how that, once we get the sequencing data out, compares with the data that you've already discussed.
Oh yeah, that'd be really interesting. And like in the case of neonatal, like to what extent are the neonatal cells? Well, you already know that they're different than the previous methods, and so like why is that? There are other questions about how much do these models that are built on in vitro data predict in vivo data. And so I showed one example of that from myocardial infarction. And a couple of years ago in JMCC, we published our first in vivo predictions looking at the hypertrophy model and showing that the in vitro based model could predict in vivo hypertrophy for some scenarios. All right, so from Angelo Calderon again, have you examined the phenotype of fibroblasts implicated in reparative fibrosis versus fibroblasts implicated in reactive fibrosis after MI? No, we have not like teased those apart yet at this stage. I think there are a few different ways in which that could be categorized. One is by stimulus. Like you're talking about like TAC versus MI, and things are going to be different. Another is like the different subtypes of fibroblasts and what area. We just haven't gone there yet, but I'm hoping that these things will be useful for that. Anonymous attendee, is there a way to incorporate subcellular localization of different receptors in the logical differential approach? Yeah, that's particularly interesting in that a lot of our older work had been specifically on subcellular signaling, and we're abstracting away from that in this framework. And so the answer is yes, if you know that detail, you can incorporate that one of two ways. One is you can kind of pretend that a protein in two compartments acts like two different proteins. Or because these are just differential equations, you can actually make a hybrid model where you have biochemical mechanistic detail for some portion of the model, and then integrate that with a larger network for which you have more abstract representation. We haven't published on that, but it can definitely work. Whether it works for your problem is that's biology. Junhoi Sun, what are essential factors needed to pay attention to transition from cell culture to 3D in situ MI tissue model with the recovery and manipulation of local microcirculation via therapeutic approach? These are things I don't know a lot about, but I'll just say briefly, yeah, there's a lot of mechanics and intercell interactions between other cell types that aren't represented in what we've been doing. And then, yeah, absolutely, things related to endothelial cells and flow and hypoxia and microcirculation are really important. Which aren't things that we've been able to tackle. Okay. Bibarani was asking if we had used a doxycycline MMP inhibitor in simulating studies. One of the things I kind of skipped over is that, yeah, we did run simulations with MMP inhibitors, like a lot of these targets. When there are cases where there are many different drugs that have the same target or targets and same binding mechanism, we just kind of lump them together so that we're only simulating sort of independent types of drugs. And so I don't remember that doxycycline is in the list. Basically, if it's in drug bank as an MMP inhibitor, then it is, but drug bank sometimes is missing known interactions, and so we've had to supplement that with manual curation. Francisco Villarreal, hi Francisco, haven't seen you in a long time. Premenopausal women are essentially protected from tissue and organ fibrosis. How does that fit into the model? What does it say about estrogens and their ability to limit fibrosis? I have no idea, and it'd be really cool to see work in that space with the models, but we just haven't gone there at all. I mean, well, there are two ways to address a question like this, though. One is if you have hypotheses, then you can incorporate those hypotheses into the model and test out what that would do. And the other thing to do is if you have no idea, then you can screen in the model for what sort of things would mimic the phenotype that you're seeing. But that's an inverse problem for which there aren't unique answers. So you'd have less confidence in something like that. Ron Vagnazzi, hi Ron. Wondering your thoughts on heterogeneity in the literature factors in your model predictions. For example, mouse strain, animal age and sex, are all relevant published studies weighted equally? Yeah, that is a interesting question. So we found, yeah, you can try to be like really specific and say, okay, we're only going to take data from a certain types, or you can be more general and say, okay, we're not going to consider those as factors. And that's the latter is what we've done so far, in which case we're basically weighting published studies equally with the standard that there have to be multiple like time points or perturbations within that same paper to that show that relationship, and that relationship is supported in an independent paper. So we're trying to prioritize where there's been some level of reproducibility. And then, yeah, definitely a lot of these experimental conditions are going to be factors, but that's where I feel looking at the model failures, or failures in quotes, because that's where we find what extent is there like intrinsic disagreement between different aspects of the literature, and we have been able to find some examples of that. But I guess I can't think of any that are quite specific enough to this that are worth mentioning. But it's in a way to figure out how consistent or inconsistent is the reported literature. And our overall finding is that things are much more consistent than we expected at first. Yeah, I mean, there are a lot of reported master regulators, but they're actually pretty related. They're not all the master independently.
So David, that reminds me of your webinar on reproducibility in science. Maybe it's not such a bad thing after all, out there.
Yeah, I'm definitely not suggesting that these reproducibility problems don't exist, but I'm suggesting kind of rather different than that. I'm suggesting that models can be a framework for assessing not just reproducibility, but generalizability.
Sure.
Yeah. Ludwig Nazis, what strikes me is that very simple assumptions about the parameters, Hill coefficient, activation curves, concentrations. Yeah, I agree. One thing that, like from that older study, we actually had original guesses, and then that were pretty good. But then because we had this really detailed biochemical model to compare against, then we could scan all the different combinations of parameters. And actually, our original guesses were really close to being optimal as default assumptions. It's that the Hill coefficient went down from 1.5 to 1.4, but other than that, we had pretty good initial guesses. But yeah, I think, yeah, surprising. Mike Kapeloff, hi Mike. How does enzyme pleiotropy and compartmentation affect model design? So in terms of the pleiotropy, there are two different aspects of the one. One is, yeah, an enzyme does multiple things. Of course, you can add those to the model. One thing, a weakness to our logic-based approach is that it does not mechanistically represent compartmentation. So it does not say, if you have a preference for substrate A versus B, that binding to A makes you less good at binding to B. And so that's, like, if you're specifically interested in that, then that's a case where the biochemical model is best for that, or at least at that portion of the network. And compartmentation, yeah, kind of similar to what I said before, you can do it phenomenologically or make a hybrid model that does it more mechanistically. Ludwig Nays again. Oh, I think I already answered that one. But maybe nature is more simple than we think. That's an interesting thing. So this is, all I can say is something philosophical about that, and it's not my idea, but John Doyle, who's a fantastic physicist at Caltech, he talks about this idea of like robust yet fragile, and that evolved systems and advanced engineering design systems look simple on the surface. They've got all this nasty, complex regulation underneath the hood. But the success of these systems is that they can look simple when you don't know what's really going on. All right, Angelo Calderon, after MI, increasing the reparative fibrotic response will lead to scar thickening and reduce LV dilation. In this regard, we do not want to inhibit reparative fibrosis, but selectively inhibit reactive fibrosis. Oh yeah, so I see a little bit more what you're going at there. Yeah, so this idea of exactly what do you want to do is to fibrosis is complicated, and that we likely want to do different things at different times in different regions of the heart. And so, yeah, I just agree that it's complicated, and we hope that these will be a tool for assessing that. For example, we've already done screens in the model to see if there's a single target that would have like opposite effects in the remote zone versus the infarct, and we don't find one so far. But I think that there's hope for doing combinations that are like two drugs that have different context-dependent effects. I think is at least an idea that has potential. Ludwig, are there examples from other fields like physics, astronomy, engineering, where such simplification also lead to reliable predictions? Yeah, so this idea of abstraction, I think, is everywhere in modeling. To me, that's what it means to model, is to abstract and to see to what extent are these abstractions or assumptions useful to prediction. It's just that it's kind of like, well, what do you mean as simple is kind of a matter of perspective, just like what you mean by mechanistic. And so, for example, like the agent-based models that have been developed, like my colleague Shane Pierce-Cotler, a lot of them are able to do fantastic at predicting spatial patterning of tissues, even if they don't have any molecular detail about the sibling networks underpinning them. So I think it's the complexity of the model needs to be paired to the questions that you're addressing. So Daniel Johnston, do you think it would be possible or realistic to link this up with a cardiomyocyte model? Yeah, I kind of already answered this, but one thing to additionally point out is that there are a lot of shared inputs and outputs between our fibroblast model, cardiomyocyte model, and macrophage models. So I think there's potential, but if we're going to do a study like that, I really want to see very outstanding co-culture systems where we can do that in a controlled way. Kishore Pasmuarthi, does your modeling work for predictions of any sex differences? Yeah, we haven't done that yet, but that'd be really interesting. And you mentioned about net flux. Is it accessible for others to help with their predictions? Yeah. So I put a link in the on the slide to on GitHub. We've posted this net flux modeling software for free. It's MATLAB based, but also we made a version of it that just has like a GUI that does not require any programming expertise at all. You literally just define a network in an Excel spreadsheet, and behind the scenes it writes all the differential equations and runs some simulations for you. And so we've had, I don't think any papers have been published just on that yet, but I've done a number of workshops with biologists that get things running rather quickly. So that's been a lot of fun. Jamie Francisco, do you think your model could be used to mimic how pathological states like obesity, type 2 diabetes, low inflammation affect the efficacy of drugs? In principle, yes, but in practice, we haven't done it an awful lot yet. But yeah, we hope so. Oh, Larry Brunton. Oh, hello. He was an amazing mentor to me in graduate school. I'll get in touch with you about IL-1 and NOS in fibroblasts. Oh, that NO can kill the fibroblasts in culture. That's interesting. Yeah, we've more looked at like NOx, and so ROS signaling in that, but we haven't looked in as much detail about NO. And some other very nice comments, so thank you for those. Have you validated this model in human fibroblast cultures or large models? From Svetlana Riley. Yeah, so some of our experiments we've done in rat cardiac fibroblasts, and some of them we've done in human cardiac fibroblasts. Interestingly, I think this probably has more to do with the passaging than anything, but our human fibroblasts are kind of quieter and have a lower baseline fibroblast activation state. But yeah, some of the TGF-beta IL-1 crosstalk that I did, we really want to see a strong TGF-beta response, and so we're using the human cells for that one. Thank you, Gillian. Nice comment. Tobias Jacoby, could you use text mining approaches for additional literature input and reduce human bias? That's a really interesting thought, and we've tried. I think I've gone back to this about every 3 years. Give another try with some really cool looking algorithm for text mining. What I find is that there's nothing to replace the manual literature curation. So my students and I read a lot of papers that every time I've tried it, there are just so many false positives from text mining algorithms. But I do expect it to get a lot better.
But I guess you probably know which papers you want to select and which ones are terrible as well, presumably. I
really try not to, but it's unavoidable to an extent. That's why we try to have objective filters on reproducibility across figures and papers and ideally labs. Yeah, it's a toughie.
Well, on that note, I think, can I just sum up on behalf of everybody, Jeff? I thought that was a wonderfully presented talk. You inspired lots of questions. You took a very complicated subject and made it very accessible to us all. And throughout it, I think the clarity of your thought processes were really coming through. I'm sure lots of people in the audience are now wondering about, can they make use of these approaches in their own work? So on behalf of everybody, Jeff, thank you very much. A couple of housekeeping things. 2 comments about next week's talks. I hope you can see my screen. On Wednesday, we keep the Wednesday, or Davor and Mike keep the Wednesday slot open for talks of special general significance. And I think nothing could be much more generally accessible and significant than the talk that Heinrich Tagmeyer is going to give. Heinrich himself is one of the world's leading authorities in metabolism and the heart, and it's therefore entirely appropriate that he's speaking about the Krebs cycle at 83, a cardiologist's perspective. And I think Heinrich himself actually studied many years ago with Hans Krebs, so I certainly recommend that. That's Wednesday. Our next event, however, is on Monday, and once again, we've got 3 talks given by people very much at the start of their careers or the middle of their careers on these 3 topics, and I strongly recommend those to you. But for now, thanks very much, Jeff. It was a great performance. Thank you.
Okay, thank you for having me. It's been fun.
No, thank you both, Jeff. It was really, really impressive. I've not seen your work, and I'm amazed. I think we need to talk a lot after this.
It'll be fun, yeah.
And David, I really, really appreciate your time again that you've committed, and for your insight, and thank you once again. Thank you for all the help that you keep providing all the time. It really is appreciated. And everyone, enjoy the weekend, and we will see you on Monday for the talk that David has just displayed on your screens. Thank you once again.